some closing remarks from EMA and FDA. If we go to the next slide, please. Let's just remind ourselves of what we set out to do with this workshop. What were our objectives? And this is just a précis from the program that was circulated some months ago. What we wanted to do was to bring together the experts and stakeholders on progressive multifocal leukoencephalopathy to a common purpose of reducing the burden of PML. We wanted to map the ongoing research and identify gaps. We wanted to find a common understanding of the priorities for research. We wanted to foster funding and partnerships and discuss mechanisms to ensure information sharing. How did we approach that? Well, we approached that with the sessions that we've had over the last quite intense 2 days. We've had an overview of PML as an adverse event. We've looked at the regulatory role, the collaborative approach, looking at how the regulators have dealt with particularly specific drug-related problems over recent years. We've talked about the treatment of drug-induced PML. We've taken stock of ongoing research in PML. We've then outlined different research agendas from different stakeholder groups. We've talked about different models for building collaboration, and we've heard about funding opportunities on both sides of the Atlantic, and particularly how you can combine collaboration with different funding models. We've talked in the last session about keeping abreast of progress of the benefits of public health. And just one thought actually on that discussion. I was struck by Panosynthesis' intervention on the HIV model. I remember as a very, very young physician in infectious diseases and HIV medicine in the early 90s, the definitive source of information wasn't one website. Different people wanted to hear from their trusted partner. And I'm sure it was the same in the US, but on the European side, generally, patients wanted to hear from patient groups. So I think the role probably of regulators and industry and others is to provide in a transparent and well thought out way robust information as well as transparency. And then that will be digested and presented for different stakeholder groups by those that they trust. And hopefully regulators will be one of the trusted parties. I take it from the interventions industry may want to be one of the trusted parties also. But I don't think it's going to be one all singing, all dancing website, because different people have different needs and different wishes. We've heard some different perspectives on a research agenda, and I missed patients off this slide, but I did during Christophe's presentation put in a last-minute patient agenda, because in fact it came in the keeping abreast discussion rather than in the discussion on research agendas first thing this morning. So forgive me for that, but it does come out. So this is just telling you what we heard before. This was the regulator's agenda. Best approach to ascertain number of drug-induced cases. So this is case definition, case capture. Is it possible to identify populations at risk or patients not at risk prior to treatment initiation with drug associated with PML? Are there early biomarkers of drug-induced PML that might be used in the mitigation of severity of the clinical manifestations of PML? What's a suitable in vivo animal model? What are the best drugs or combinations of drugs with anti-JCV activity? What are the best treatment strategies for both PML and IRS? What's the long-term safety and effectiveness of plasma exchange and immunoabsorption? What is the best tool to evaluate the effectiveness of risk minimization activities in PML? That's what the regulators were saying coming into this meeting. In terms of academia, this comes from the presentation this morning from Jean Major. Research approaches in terms of molecular genomics, proteomics, viral and host, viral gene regulation in specific cells, be they glia or neurons, immunology of therapies and host response to infection. And then in the area of drug discovery, small molecules for the interruption of viral growth once identified, relevant cell models for screening, vaccines, peptides, VLP like papilloma, prophylactic and therapeutic, clinical studies, relevant animal models for pathogenesis intervention. If we then moved on to the clinical agenda, and you'll see there's a lot of commonality in these different perspectives on the research agenda. But risk, and sometimes it's the same question being put a different way. And I think that's actually quite a useful validation in terms of our overall agenda for the next of this workshop. So risk-benefit ratio for treatment: should my patient be treated with drug X? Predictors for PML, which patients should not be put on the drug, what are the lab parameters I should be following in my patients treated with the drug, MRI, CSF testing, etc. How often to do that testing? What's the value of a drug holiday? Should I stop the drug after 12 months, 18 months, 2 years? PML diagnosis in, for example, an MS patient who's experienced a new neurological problems. How do you distinguish PML from multiple sclerosis relapse? How to treat PML, how to mitigate risks through patient selection. If we then, the industry colleagues presented it in a part as things they had already identified to fund, as well as things to be funded in the future, but I think it equates largely to a priorities for research agenda. And the high-level agenda was to better predict, prevent, and treat PML, creation of a clinical database, gathering information on demographics, clinical information, and brain imaging, establish a repository of samples, funding of academic collaborative research network, and that looked a lot of basic biology of JCV, pathogenesis of PML, immune response to JCV, viral changes associated with PML, animal models of PML, clinical validation of risk stratification assays, and clinical evaluation of emerging PML therapeutics. Now, Christophe, this next one I put in nanoseconds ago, so hopefully I captured it correctly. More collaboration on benefit, risk, methods, and decisions. Collaboration, I think, particularly with patients, was your point. Improving the pathways to disseminate information on benefit risks and on PML more generally. Clarifying the role of antibody as risk indicator. Risk of PML before the 3 years. I think that was rather specific to natalizumab, but I think we could broaden that to the risk of PML at different stages of treatment with different agents. Development of anti-JCV therapy. So, I then hand over to Gerald.
Okay, so we've brought a lot of people here from a lot of different stakeholder groups, and we've seen everything from gene maps to discussing things such as how medical care systems should work. And you have to bring all that together if you're going to do something like we're trying to do here, deal with a rare but serious adverse event. So we've brought the key stakeholders together in an attempt to foster capacity building through networking, information sharing, best practice, tackling obstacles, and collaboration. What we said in the beginning is even more so true today, and what many people have said, none of us can do this alone. And so we support a PML research agenda that really has the patient as its primary focus. And we, like everyone else, encourage the dissemination of research agenda to funding bodies. And go down here. And so what would the immediate next steps be? Well, first, to make the proceedings of the conference available on a website and a journal, and that the research agenda would be disseminated to funders. We at FDA are not funders of this type of research, and encourage future research collaborations. And the conclusions, we as regulators have provided the forum here for people to meet, to exchange, to discuss ideas. Now it's up to everyone here to play our own part, to treat the patients, to prevent preventable adverse events, to diagnose disease properly, to lead to proper treatment, and to diagnose adverse events properly as well, to continue research in these areas, and to collaborate. That I can't underscore that word enough. For those who have the ability to fund this work, and for all of us to share the knowledge we're getting, and to a common purpose of reducing the burden of PML. So I will turn this back over to our host from EMA.
Just in, you know, they're filming, so it's just so that we don't cause confusion of identity at future. When somebody in a few years' time has got a quiet couple of days and they look at the webcast and work out this man Gerald and this man Peter, who seem to be interchangeable. It's summed up on the screen. Thank you. I mean, first of all, thank you to FDA colleagues, all the colleagues that have come over, contributed to this workshop. Gerald in particular for being very much the stimulus for the idea of a research agenda and a workshop. Thanks to EMA colleagues, particularly Monica Venus, who did a lot of the scientific legwork for this conference, who unfortunately couldn't be with us today for family reasons. Ava Rossi, who has done a sterling effort from an organisational support, along with Valerie Muldoon, Henry Fitt, and our colleague Manuel Haas, who's also made a major contribution. I'd like to thank, on behalf of EMA as your host, and probably on behalf of FDA, if I'm allowed to speak on behalf of FDA as the co-chair, thank all the co-chairs, all the speakers, all the participants, all the probing questions, because I think it's been really interactive and we have moved the debate forward significantly in the last 2 days. So thanks to you, and particularly those that have come a long way to come to this workshop. We all have a common goal. The common goal is reducing the burden of progressive multifocal leukoencephalopathy, and it is clear that if we collaborate, if we remain focused, we can make the situation better. So have a safe trip home. Thanks for your collaboration, and things will get better.